Welcome to my channel. Today we are going to discuss about Cambridge International AS and A levels biology paper 1 multiple choice. It is the paper of May-June attempt 2022 and the required time to complete the paper is 1 hour 15 minutes. So what you need to do, you need to just adjust your speed according to the timing. There will be 40 questions and all the questions are compulsory. Question number 2. Four students were asked to match the function with the appearance of some cells structure in animal cells. The function were listed by the number. Messenger RNA passes through the ribosome, produces the mitotic spindle during cell division, packaging of hydrolytic enzyme that will remain in the cell. The appearances were listed by the letter V. Membrane which surrounds an enclosed inner cavity. W. Non-membrane bound spherical structures. A double membrane interspersed with pores. Membrane bound sacs arranged as a flattened stack. So these are the structures and you have to match with their functions. Which student correctly matched the numbered function with the appearance of the cell structure? So messenger RNA. Messenger RNA passes through to the ribosome. A double membrane interspersed with pores. Nucleus. So we are talking about here the nucleus. This is the nucleus. It is a double membrane structure and it has pores. It has pores. From these pores, messenger RNA passed towards the ribosomes. Produces the mitotic spindle during cell division. Cylindrical structure, centriole. We are talking about here the centrioles. Packaging of hydrolytic enzyme that will remain in the cell. Membrane bound sac arranged as a flattened stack. So the answer is D. Answer is D. Which size range would include most prokaryotic cells? If you can see here, this size range, B. Prokaryotic cell, they are mostly in micrometers. And you should also take a look at the brackets here. Never miss your diagrams from the book. Such questions are being asked by the examiner from the diagrams. What is present in a typical prokaryotic cell and a typical eukaryotic cell? So we must talk about the similarities between them. Centrioles can never be present in a prokaryotic cell. Circular DNA is not in the eukaryotic cell. Starch granules. Animal cells don't have starch. Plant cells have starch. So and prokaryotic cells also don't have some prokaryotic cells which are photosynthetic in nature. They may have starch granules, but 70s ribosome is a must. So the answer is 4. The answer of 4 is A, 70s ribosomes. Question number 5. Which statement about viruses is correct? They all contain RNA. No, they may have a DNA as well. They may have a DNA as well. They all have an outer envelope made of phospholipids. Phospholipid is a cell membrane. They don't have a cell membrane. They have a protein coat. Never confuse it with the protein coat. They all contain ATS ribosomes. ATS ribosomes, of course not. So they have a capsid made of protein. This is the right answer. Question number 6. Samples of glucose, sucrose, and a mixture of glucose and sucrose were divided into two halves, M and N. M was then tested with the Benedict solution. N was boiled with dilute hydrochloric acid, neutralized, and then tested with the Benedict's solution. The color of the solution was compared to color standards. Which table identifies the correct color changes for these samples? M is the Benedict, N is the dilute hydrochloric acid. Okay, M is the Benedict, N is the hydrochloric acid. So, if we talk about the Benedict solution, reducing sugars give the positive test results, and the non-reducing sugars don't give any result. Benedict solution is blue in color. Sucrose is not supposed to give any color. So answer is C. Yes. If you compare all the tables, how it can be C? Glucose turned the Benedict solution into yellow, and of course the end result of the hydrochloric acid will be yellow. Sucrose didn't give any result, which is, you can see there is blue color, and sucrose gave a same yellow color with the HCl. The mixture of both gave yellow color to the binding solution. Because of the glucose, the color of the binding solution changed, and the HCl is turned into red. HCl will turn into red. You must do the test for the sugars and test for the proteins and lipids. So this question is from your practicals. You must also do a very good uh, overview of all the practicals as well. Question number 7. Which molecules contain 1,4 glycosidic bonds? 1,4 glycosidic bonds. You must also remember the glycosidic bonds of all the important carbohydrates. Answer is C. Glycogen and cellulose. They both contains 1,4 glycosidic bonds.